These are your notes on the structure of the cell membrane and the function of the different parts of the cell membrane. This is the overview of the cell membrane, so most of this information you should probably have to write down.
Okay.
A lot of this is new vocabulary. And I do want to say hello to my kids. Say hi.
Hi.
Okay, so as far as the overview goes, the function of the cell membrane is that it separates the living cell from the non-living surroundings. It's very thin. It's 8 nanometers thick, so that's a very thin substance. Its function is that it controls traffic in and out of the cell, or it regulates the cell. It is said to be selectively permeable. That's a vocabulary word for you to write down. So what that means is that it allows some substances in more easily than others. And we've talked about that. We've talked about how if a substance is water soluble, it has to use a protein to get through the phospholipid bilayer because the tails are hydrophobic. But if it's lipid soluble, it's able to just diffuse directly through the membrane because it's made up of phospholipids. You should also know that it is made up of phospholipid proteins and other molecules such as carbohydrates and cholesterol.
Do the whole thing to make sure your speech is perfect.
We've talked a lot about phospholipids in class. If you don't have this information written down, you should write it down. And so this is what it looks like in terms of a chemical representation of a phospholipid. So it has the head is going to be hydrophilic, meaning water loving, and then the tails, so here are the tails on the chemical representation, are hydrophobic, meaning water fearing. And so the way that they orientate themselves is so that the heads, this is inside of the cell, and this is outside of the cell, then the heads are going to be exposed to the water that is inside and outside of the cell, which is fine because they are hydrophilic. And then the tails are hydrophobic, and so they are what actually regulate the cell and prevent water-soluble substances from diffusing directly through them according to their concentration gradients. So this is just a slide that shows you what the phospholipid bilayer actually looks like. So you can see the heads are outside and inside the cell, and then the tails are part of this bilayer section. And they will do this automatically. Sometimes part of the cell will pinch off or form around other molecules, and this phospholipid bilayer will automatically form like this. In 1992, Singer and Nicholson proposed that the membrane proteins are inserted into the phospholipid bilayer. This is when the whole idea of the fluid mosaic model came into play. We talked about that in class and how a mosaic is a piece of art that's made up of lots of different pieces that come together to form one art piece. That's the same thing for the cell membrane. It has this phospholipid bilayer. It's got all kinds of different types of proteins. And they all come together to form this one organelle called the cell membrane. So this is just showing you a bunch of different types of the cell, or the different parts of the cell membrane. We've talked about these in class. This is illustrated probably better than what I was able to draw for you. But here we have the extracellular fluid, so that's outside of the cell. Here we have the cytoplasm, and so that's inside of the cell. Whether it's outside or inside of the cell, it's water. And so we can see our peripheral proteins, which are proteins that are only located on one side of the cell. They don't go all the way through. And we have transmembrane proteins, which go across the membrane. So those are the ones that go all the way through. So you should write that down, and that's actually going to be coming up on the next slide, give you a better definition. But transmembrane proteins go all the way through. Peripheral proteins only go on one side. Cholesterol, we talked about how cholesterol is found within the tails of the cell membrane and how it helps to give it some structure. glyco, hopefully you'll remember that from our macromolecule unit when we were talking about glycogen. So this is the carbohydrate attached to a protein. And we talked about how, or I mentioned in class, how this is used for cell-to-cell signaling. So lots of different parts of this mosaic. So this is the slide that gives you the definitions of the protein, so you should write this information down. The proteins determine the membrane-specific functions, and so we've talked about how with all proteins, it's their shape that determines their function, and that's the same thing here. The cell membrane and organelle membranes each have unique collection of proteins, so no two cells are going to be the same. If they're making up the same tissue, then they're going to have the same similar proteins, but their sequencing of proteins and stuff is going to be different. The different types of proteins, they can be categorized into two main groups. So you have your peripheral proteins, which again are going to be bound to one surface of the membrane, so they're going to only be on one side. They're not going to go all the way through. And they can have a couple of different functions, but one of them is as a surface identity marker, or that's referred to as an antigen. And so they help cells talk to other cells and signal the health of the cell that they are in. The second major category of proteins are called integral proteins, and these are transmembrane proteins, meaning they go all the way through the cell. So if you look at this picture right here, this would be a transmembrane protein, that would be a transmembrane or integral protein. They are generally going to be transport proteins, and so their function is going to be to get molecules from one side of the cell to the other side of the cell. And they can be channels. They could use, sometimes they use pumps. And so this would be an active transport, so it requires ATP. Carrier proteins are transmembrane proteins as well. So lots of different types of proteins, but two main categories: peripheral proteins, which are only on one side, integral proteins, or transmembrane proteins that go across the whole membrane. So you don't need to write these down, but this slide is just to give you a visual of the different types of proteins. And so these are going to be all transmembrane proteins, and so you can see that they're all going from one side of the membrane all the way down through the other side. And so we have a transporter protein. So it's just allowing for these red molecules to diffuse through it according to its concentration gradient. So here we have high concentration, here we have low concentration, and those molecules are able to diffuse from one side of the cell membrane to the other side of the cell membrane. This would be an enzyme type of protein. So we talked about enzymes when we were studying proteins. And so this substrate comes into the active site of the enzyme, it gets changed, and it gets released as a new molecule. This is a surface receptor, so it's able to receive molecules and then deposit them on the inside of the cell. This one down here is an identity marker, again, so it's got that glycoprotein attached to it, which helps in cell-to-cell identification and the cell being able to start an immune response if the cells need to. We know that when cells stack upon each other, they form tissues, and then tissues go on to form organs, and organs form our organ systems. And so the way that cells stick together to start to form these tissues is through cell adhesion. We talked about adhesion, and that's when things stick to other things. And so here we have one cell sticking to another cell. And then attachment to the cytoskeleton. So we haven't talked about cytoskeleton yet, but the cytoskeleton is kind of like the skeletal structure of the cell. And so it's what gives the cell a three-dimensional structure, and it allows so it prevents the cell from collapsing on itself. And so the proteins themselves will attach to the cytoskeleton to try to gain some support and structure for the phospholipid bilayer as well. This slide focuses on the membrane carbohydrates. So you need to know this, so you should write this down. The main role that they play, again, is, we've talked about this, is the cell-to-cell recognition. So allowing one cell to talk to the cell that's next to it, and say, okay, I'm a kidney cell, you're a kidney cell. We're going to adhere to each other with our adhesion proteins, and we're going to start to form some kidney tissue. The second thing, so that is important in organ and tissue development. And then another major thing that the that the carbohydrates do, or the glycoproteins do, is it sparks the immune system. And so if a cell comes in and its glycoprotein doesn't match the glycoprotein of a cell that's next to it, then that will start up your immune system. And so it's your body's way of identifying foreign cells, or cells that should not be in your body. And so if you have a tumor growing, for example, your body will start to identify that it's a tumor cell and not a kidney cell, because the glycoprotein that's attached to the tumor cell is not the same as the glycoprotein that's attached to your kidney cell. And so carbohydrates play a major role in cell-to-cell talking and just the overall health of the cell.
Bye, class.
Hold on, it's recording.